I'm Daniel Levine, and this is the Bio Report. One problem with drug discovery and development is that answers about the way substances act in the body are often not apparent early in the process. Though human cell assays have been used, they have their limitations. BioSeek's BioMap technology seeks to improve the success rate of research and development by bringing the complexity of human biology to cell assays and incorporating that into the earliest stages of the drug discovery process. We spoke to Ellen Berg, Director and General Manager at BioSeek, about the company's technology, how it seeks to improve discovery and development, and other ways that technology is being used by the industry. Ellen, thanks for joining us.
Well, thank you.
One of the reasons that drug discovery and development is so expensive is not only the high rate of failure, but the fact that failure often comes after there's significant investment in a potential drug, only to find there's a safety risk. How are drugs typically screened before they enter human trials, and what are the limitations of traditional approaches?
Yeah, there is a lot of work that's done on any compound or new drug before it gets into people. There's in vitro assays where cells are in culture, animal studies to try to pick up effects in a whole organism, and it's years and millions of dollars invested before we even start clinical trials. So the types of assays are usually a simple cell culture with a cell line or a liver cell, and looking for overt adverse effects of a compound or a drug. In vitro, looking simply at cell death, and then in vivo, giving animals large doses of the drug and looking for, just any kind of phenotype, whether loss of blood pressure or, the animal starts shaking or things like that. And so, the assays we have had up to now have been fairly crude and frankly not predictive, and that explains why we haven't done a better job of reducing attrition in clinical studies. Over the last 20 years, it's still hovering around 30% of clinical failures are due to unexpected toxicities.
Can you explain what your BioMap platform is and where it sits in the discovery and development process?
Yeah, so it is a in vitro approach that is aimed at bridging the gap between whole organisms and cell culture. So we decided that we're in most the cell in vitro assays that were available, that are available to us now, they're too simplistic and they don't do a very good job of modeling tissue biology or organism biology. And so we went back, to the beginning and said, what are we missing? One of the easy things that we're missing is the fact that cells talk to each other. And so a lot of our assays have co-cultures where we have more than one cell type, together in a dish. The second aspect is the kind of cells that we use. We use primary human cells. So they're human, they're from people, and they are taken from normal donors and not subject to genetic engineering or long-term culture in a dish, so that they are more like cells in our body. So they retain a lot more of the normal regulatory controls that cells do when they're in our body. People may not realize that most of the in vitro work is done with cell lines, which have been propagated in cell culture in plastic dishes for years and years and years. And the famous example is the HeLa cells, which have been in culture for something like 50 years and are essentially Franken cells. They've lost a lot of the regulatory pathways that normal cells have, so they're not representative of normal cells. So we use primary human cells, we use co-cultures, and the third aspect is we control the disease environment. And so we create little models of tissue disease in a dish by adding in factors to supplement what the cells might make, to make them feel as if they were in a tissue or in the body. And then what we measure are actually disease risk factors. So clinical biomarkers that are already known to be important for disease. So with these three aspects of the, assays, coupled with the fact that we designed these from day one to be high throughput so that we could generate very large data sets and compare many, many, many drugs and compounds to each other. So, that becomes, a real powerhouse of information that, helps you, first of all, understand, are these assays predictive at all? And, do the experiment. And over the years, we've built up more and more validation and have shown that for many aspects, these assays are showing good, predictivity of effects of drugs, in the clinic, whereas other in vitro assays have not, don't have the degree of predictivity.
So, in designing this platform, what were you seeking to do, and what were the challenges in doing it?
Yeah, it started with a very simple observation that in my little narrow area of research, which was vascular inflammation, I had been looking at disease biomarkers that in vitro cultures that everybody was using at the time, their expression, the regulation of their expression, they would increase in expression and then go, the time course of that was very acute, and it would, the level of that biomarker would go up and then go down and disappear very quickly. But yet I knew in vivo that this biomarker got upregulated and remained very high expression, for months. And so I knew that something in vitro we were missing. So what we were missing is, other factors that we added and then could recapitulate, the regulation that we see in vivo. So it was the observation that what we saw in vitro wasn't matching, and trying different things. What are we missing? Adding in either other cells, right, as a stimulus, so to speak, or other factors that we knew were present in the tissue in disease. And the gold standard was, what does it look like in people? So matching the phenotype in people, that was really the key.
So it seems like there are two issues here you're trying to get at. One is the complexity of biology, and
Yeah.
the other is kind of that industrial high throughput screening. How successful have you been at first mimicking those disease states, and then when you're doing it in a high throughput way, how much data can you generate? Is there a way to quantify that?
Yes. So I would say that one of the novel aspects of what we were trying to do, or the more difficult aspects, I would say, is combining biologists with computational people. So, the informatics plus the biology, usually those types of researchers don't speak the same languages. So getting them to work together and, being able to generate very large data sets where you can really test your ideas in a very critical critical manner was challenging, but certainly turned out to be very advantageous for us. There's no limit really to the number of compounds and assays you can do. There have been huge advances in miniaturization of all kinds of in vitro assays, and we've taken advantage of that. And multiplexing, we can now get so much information out of a very small, sample. So that has really opened doors and opened, certainly made these capabilities, available. So there is no limit. Of course, there are practical limits in terms of numbers of compounds you could test or agents you could test, but the most interesting things, there's not that many interesting things to test. I'll, be frank. One of the things that we struggle with or we're interested in are, for example, very selective probes for maybe that would be inhibitors of a specific target. And, those are few and far between. There are a lot of non-specific probes or test agents that one can look at, but they're not very informative. So there's not that many drugs, right? The FDA-approved drug list is like 1,000. That's nothing.
So are you able to answer questions that previously could only be answered by putting drugs into humans?
Yes, and more than that is we help explain why a particular clinical activity is seen, either an efficacy or safety-related effect. We certainly don't cover all of biology, and I wouldn't claim to even, claim to do that, but it turns out that any additional information, even 5% better than what we have now is still $1 billion proposition, given the difficulties that we have. So, and we're continually expanding the platform. We cover a lot of tissue biology, inflammation, cardiovascular, respiratory. We're expanding into kidney and fibrosis, oncology. CNS might be the next area that we'd like to, expand the technology. There are limitations to everything, and so what we do certainly fits in, is complementary to other types of studies. And the nice thing about what we do is that it's a bridging technology. It bridges information at the molecular level all the way up to, what we're measuring in clinical studies. So because of our focus on translational biomarkers as endpoints in our assays, that really bridges that gap. So it's a nice technology that fits and connects a lot of other approaches that people are interested in using.
And is there any data on time or cost savings using the technology?
Yes. One of the more interesting areas right now in drug discovery is interesting approaches that people are going back to is phenotypic drug discovery, which is, testing compounds in a live biological system, and without really knowledge of the molecular target that you're screening against, and that has been a very rich source of new drugs. A lot of first-in-class drugs were actually identified through these types of, whether they were animal models or even observations with people. And then the target was subsequently discovered. But there's interest in, doing phenotypic drug discovery. But when you do that, you get a compound or molecule that has activity that you want, but you don't necessarily know the molecular target. And there's a lot of concern about, wow, if we don't know the molecular target, that adds a lot of risk, because what if it's something known? What if it's something that we know has side effects? And what about how do we protect the intellectual property? And so one of the things that we can do is automatically classify molecules as to their target and pathway mechanisms of action based on all the known molecular targets and compounds and tool compounds that we have in our database. And so what we're able to do for folks in this area is rapidly triage out known mechanisms from something that might be novel, and also activities that might have safety issues. So that can save people a lot of money. We had one of our clients told us that they had some great compounds that were effective in animals. They were identified in a phenotypic screen, and they had, gone all in with this program, gotten a lot of animal data, and spent at least a year trying to identify the target. And they said that they had spent, $1 million outside, without external, contractors trying to identify the target. And with ours, it was a matter of 6 weeks and fewer than three-figure or fewer than a six-figure dollar value, and they immediately got the answer. So we could have saved a lot of time and money on those types of projects. In other cases, we actually, for safety, rather than killing projects, we've actually been able to rescue projects by identifying why was there a safety issue, and which helped actually manage the program and continue it in clinical trials. And I think that's more valuable when you can rescue a program.
We talked about complexity, but human variance is also an issue. We're in this age where developers are becoming very conscious of personalized medicine. And if you put a drug into 100 patients, they're not all going to react the same way. Does your platform allow for variance in any way?
So, yes, it does. One of the interesting things about human variation and in clinical trials is the fact that exposure levels among patients, among individuals who are receiving a drug, vary by tenfold. So that's one of the things you can assess in vitro is the therapeutic window, so to speak, which when you get into the population you need to have a good window between efficacy and safety to allow for that variation in exposure. So that's number one. Number two, in terms of looking at how individuals respond, we have actually done customized studies with our platform where instead of using it as a screen for new compounds or for mode of action type of thing, to actually look at, what is the variation in response. And so because you can do individual donors, you can pool them, you can, mix and match, that sort of thing. You can tie it to genetics, right? Now, the challenge with that is the number of donors you need to look at. And the reality is, even the folks who are doing, GWAS analysis, the, genome-wide association studies, understand is there's not enough people in the world to really, there's not enough samples, shall we say, to really do the kinds of studies we would like to do in trying to associate genetic differences with, particular effects. So there's genetic components and there's also environmental. So I think we certainly contribute to our standard platform contributes to environmental differences. And that's when you see we do combination studies, so you can look at drugs together. And we know that when we go into the clinic, some people, for example, in arthritis, they will be on a baseline therapeutic like methotrexate, and some people won't. So there's variation. So I think variation takes a lot of flavors, and all of that could be addressed with what we do. Sort of depends on the question, but it's certainly doable in an in vitro format.
You've got a number of partnerships from drug companies to the U.S. Environmental Protection Agency. You talked a little about the ways that the technology is used, but can you walk through maybe a few of these relationships to illustrate the different way the technology is being used?
Yes, and what the EPA is doing, which was a very interesting program that they spearheaded now about 8 years ago, was they were look for in vitro assays, high throughput assays that would help them prioritize chemicals of most concern. They have a 10,000 chemical backlog for toxicology studies. And there's just not enough time or money in the world to do full tox programs on all of those chemicals. And they needed a way to prioritize, which ones would be the most concerning. And so, what ideally for the EPA and their stakeholders, which are chemical companies and environmental folks, and you and I, people who are exposed to all of these things in our daily lives, is a rapid way to tell which compounds or chemicals might be of concern. And so, these in vitro assays, provide you with that. Part of what the EPA was doing was actually doing a bake-off of in vitro assays and correlating the data with in vivo studies and traditional toxicology studies to see what types of in vitro assays would be predictive of adverse in vivo effects that they could use to triage. And they've gotten some assays and they're moving forward with that. So everybody's happy there because you want to use as few animals as possible, protect the animals, and you want to get better data, and you need to do it faster and less expensive. So if in vitro approaches are sufficiently predictive, that's a no-brainer. So that's what the EPA is doing, and now their project has now become part of this bigger effort at the NIH, the Tox 21 program, which involves FDA and NIEHS and NTP, National Toxicology Program, and the full NIH for expanding this approach, not to pharmaceuticals and to drug discovery. So that's ideally that's where we'll be with that effort. Someday we'll be using in vitro assays in regulatory to to qualify how safe is your compound. The other types of things we're doing with pharmaceutical companies come to us with all kinds of problems, issues, and projects. They might have a compound they don't understand why it works. They might be in a very highly competitive target-based program, and they need to distinguish their molecule from competitors. And those are the kinds of things we do. We also use our assays to do drug discovery. So we've run high-throughput screens with individual assays that we've either run in-house or we've transferred out to run a high-throughput screen and find, new chemical matter and, starting drug discovery programs from scratch. Using this phenotypic drug discovery approach I talked about. So those are the kinds of things that people come to us, and they usually are coming to us for biology that they don't have the time or inclination to develop in-house. So that covers a lot.
Well, there's also been some efforts to repurpose drugs and rescue drugs that may have been abandoned in clinical development for one reason or another. Does your platform have potential in that regard?
Yes, we've done quite a few studies on this regard, and those are very interesting. There's usually some degree of clinical information, and the goal is to either find a new indication or to understand the mode of action so that a new indication can be developed. So we certainly do those. I think the challenge is more on the business side for the folks, that we do this for is, how do you craft either intellectual property or, a path forward, because molecules that are off patent, it's hard to get funding to, further, develop this in terms of the clinical trials, without them, being able to, at some point, create a product to make money. So that's the challenging part. But I think, it's certainly very interesting use of our platform.
Any sense how the FDA looks at the validity of findings from this approach or data generated by it?
Yeah, I think so far they've seemed to like it because it has, for the INDs that have been, our data has been submitted, and we actually write quite a few IND reports that go into submissions. That it provides that link that I mentioned between other data, in vitro data or animal data, and the clinical endpoints. And I think so far what I've heard is they appreciate it because it makes sense. It's actually easier for clinicians to understand our data. It's than, for example, a whole genome, microarray profile where they probably are crossing their eyes. Too much data, not enough information. Knowledge, I would say, and ours is more of a how it is used, by the researchers is to explain things. So to pull everything together. So that's they seem to like that and appreciate that.
And you don't use big words.
Right.
Ellen Berg, Scientific Director and General Manager at BioSeek and developer of the BioMap technology. Ellen, thanks so much for joining us.
And thank you. Appreciate the time.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.